Hi everyone, this video is going to be talking about cell membranes and transport. So we'll start by looking at sort of the basic structure of a cell membrane, and then we'll get into the movement of molecules across that membrane, so the transport side of things. Okay, so we're going to start by looking at the basic structure of a cell membrane, and there are two things in particular that I want us to pay attention to. So, one component of a cell membrane is that it's composed of a phospholipid bilayer. And these phospholipids are shown here in sort of red as the head and these yellow tails. And these phospholipids, as their name implies, they're a kind of lipid or fat. And their main job is to prevent the movement of a lot of different kinds of molecules and ions from crossing the membrane. So they're sort of guards or barriers. So the cell membrane is going to have this phospholipid bilayer. Bilayer is going to refer to the fact that we have two layers of phospholipids. So you can see there's one phospholipid here with the head facing the inside of the cell, and another one here with their head facing the outside of the cell. So, sort of like a sandwich with the two slices of bread and filling in between, we're going to arrange these phospholipids in a bilayer. And then the second component that I want to highlight are these proteins. And these are going to be embedded within that phospholipid bilayer. And these are going to be structures that will allow specific molecules to come through and either exit or exit the cell. So they're going to be regulating transport, but in general, allowing controlled movement of cells across the membrane. Okay, so our next slide is going to sort of focus in on what a phospholipid is and structurally what does it look like. So as I mentioned before, they're part of a lipid family. So that's going to include other molecules like fats and oils. And one individual phospholipid is going to have a few different parts to it. So this has the same coloring as what we saw in the previous slide. So the head of the phospholipid is going to contain one molecule of glycerol and one phosphate. So that's the red portion here. And these heads are going to be hydrophilic, and that means they like to be around water. They're water loving. And then the other part of a phospholipid is going to be the two fatty acids. So that's this yellow structure here. And so these are the fatty acid tails, and the tails are what are called hydrophobic. And this means they're water hating or water fearing. And when we're thinking about the structure of that phospholipid bilayer, you can see now that the water hating tails are sort of hidden away on the inside of the membrane, and the water loving or hydrophilic heads are going to be protecting them sort of on the outside. So the watery cytoplasm on the inside of the cell is going to be in contact with this hydrophilic head, and there's water on the outside of the cell too. So the hydrophilic head of the outer layer of phospholipids is going to be protecting them as well. So the structure of the bilayer is going to be oriented such that the hydrophilic heads are shielding those hydrophobic tails. Okay, so I next want us to think about what kinds of molecules are able to cross the cell membrane and which kinds of molecules will need a protein to sort of help them across. When we're thinking about the kinds of particles that are able to move across that lipid bilayer and the kinds that are going to need a protein, it's really going to depend on the size of the particle as well as whether the particle is charged or not. So let's take a look. Let's take a look at a diagram of that. Okay, so here we're showing that smaller and uncharged molecules are going to be able to move through the phospholipid bilayer without any help. So, an example is oxygen or carbon dioxide. These molecules are really small, and you can sort of imagine that it'd be easier for a small molecule to sort of squeeze through this phospholipid bilayer. The other important thing is that they have to be uncharged. So, they can't have any pluses or ions. We're not talking about any sort of ion, they have to be neutrally charged. So small and uncharged molecules will be able to get through. However, larger molecules, so for example, glucose you can see is much larger, and any sort of polar molecular ion. So amino acids are often polar, and that just means they have little regions of sort of partial charges on them, or a molecule could be fully charged. So an ion would be an example of that. If you see a little plus sign, +2, or a minus sign like this chloride ion, these molecules are not going to be able to move through the lipid membrane. So, when we're thinking about how they'll get across, if we need to move them into or out of the cell, they're going to need a channel protein or a transporter, so some sort of helper protein to enable them to cross. When we're thinking about molecules moving across the membrane, this idea of diffusion is going to control which direction they move in. So diffusion is saying that particles will move from regions where they're in a higher concentration to regions where they're in a lower concentration. And this is a passive process, so it doesn't take energy. It will occur spontaneously or automatically. You can see this with the dye here. So if you drop food coloring into a beaker of water, the dye will be concentrated, highly concentrated originally in sort of this drop, and then it'll move from that high concentration area to an area of lower concentration. So from high concentration to low concentration. So you can think of particles as being a little claustrophobic, maybe. They don't like being in areas where there are a lot of other particles of their same kind. So we'll first look at two different kinds of diffusion across a cell membrane. So, when particles diffuse across the cell membrane, they're going down their concentration gradient, going from regions of higher concentration to lower concentration. And there are two ways this can happen. One way is called simple diffusion, and that's when you don't need any sort of protein. So, the molecule is just going to move straight through the lipid membrane itself. And so, this means that they're going to be small and uncharged molecules, and they're just going to move right through without the help of a protein. Facilitated diffusion is when the molecule is maybe just a little too big, or maybe it has a charge, but for whatever reason, it can't cross the membrane by itself. So it needs a little bit of help. It needs to be facilitated with the assistance of a channel or a carrier or transport protein that's going to enable it to get across. So particles are still going to move down their concentration gradient, but they're going to do it with the help of a membrane protein. So diffusion can be either simple diffusion or facilitated diffusion, but in both cases, you're going to go from high concentration to low. So it's still, they're both examples of kinds of diffusion. And this is the direction that molecules want to move in. They want to go from high to low, so you don't need to put in any energy to make it happen. So it's passive. The next type of transport that we'll talk about is called active transport. And in this case, we're moving particles against their concentration gradient. So we're moving particles from regions of lower concentration to an area where they're higher in concentration. So if you look at these squares, they're at lower concentration on the outside, and we're pumping them into the inside where they're in a higher concentration. They don't want to move in this direction, so we have to use energy in order to make this happen. And active transport's always going to need a transport protein in order to make this happen. We can't call this a diffusion because we're moving from low concentration to high concentration. So the molecules aren't moving in the direction that they want to go. They're not moving down their concentration gradient. And it's active because it requires that cell to use energy in order to make it happen. So this is just a review slide showing both kinds of transport next to each other. Again, we have two different kinds of passive transport, simple diffusion, where we go straight across the membrane, facilitated diffusion, where we're using a carrier protein or a channel protein. But in both cases, for passive transport, we're moving with the concentration gradient or down the concentration gradient from high to low. Active transport, we're using energy and we're moving molecules against their concentration gradient from low concentration to high concentration. Okay, in this last slide, we're talking about two systems of transport for molecules that are just way too big to get in. So they definitely can't move straight through the membrane, and they're too big for even a carrier protein to be very useful. So the two ways that we can get molecules in or out are called endocytosis and exocytosis, respectively. So both of these kinds of transport use membrane sacs called vesicles, and we're going to use these vesicles to move the cargo in and out. The picture on the top shows a process called exocytosis. So, in exocytosis, we have inside of the cell this vesicle. So, we have a little mini membrane sac, and inside the vesicle, there's some sort of cargo that we want to be transporting out of the cell. And so, the vesicle is basically going to fuse with the cell membrane and then release the contents of the vesicle to the outside of the cell. So we're exporting cargo from the inside of the vesicle to the outside of the cell. So export, exocytosis. The opposite process is called endocytosis, and that's where we're bringing content into the cell. So you can see these molecules here. And the cell membrane is going to sort of wrap around that cargo and sort of enwrap it, encircle it, and then it'll sort of pinch off, and we get a vesicle now on the inside of the cell, and we have that content that used to be on the outside now on the inside of this vesicle. So exocytosis to export cargo, endocytosis to bring material into the cell. That's it for our video lecture on cell membranes and transport. So I'll see you guys soon.